welcome to SAM Biology class. Friends, today we shall discuss about the tertiary structure of proteins. Before that, I request all of my friends, those who have not subscribed my YouTube channel, please subscribe. The tertiary structure of a protein describes the folding of its secondary structural elements and specifies the positions of each atom in the protein, including those of its side chains. Secondary structural elements are formed due to the folding of short contiguous segments of the polypeptide chains. Alpha helix and beta sheet are the two most common secondary structures found in proteins. Others are 310 helix, pi helix, random coil, coil and different types of loops and turns. So these secondary structural elements are folded in specific pattern to form the tertiary structure. Around 110,000 protein structures have been reported. No two are exactly alike, but they exhibit remarkable consistencies. The primary structure of globular proteins generally lack the repeating sequences that support the regular conformations seen in fibrous protein. However, in globular proteins, amino acid side chains are spatially distributed according to their polarities. Number 1. Non-polar amino acids such as valine, leucine, isoleucine and phenylalanine occur mostly in the protein interior where they are out of contact with the ecosolvent. The hydrophobic interaction that promotes this distribution is largely responsible for the three-dimensional structure of the native protein. Number 2. Charged amino acids such as lysine, arginine, histidine. These are the positively charged amino acids, and negatively charged amino acids such as aspartic acid and glutamic acid. Both positively and negatively charged amino acids are generally found on protein surface, but the non-polar amino acids are found in the protein interior. Number 3. Polar amino acids such as serine, threonine, asparagine and glutamine are usually found on protein surface, but sometimes they are also located in the interior. When buried in the protein, polar amino acids always remain hydrogen bonded to other groups. Loops and turns that connect secondary structural elements are also present at the protein surface. The general principle of side chain distribution is evident in the individual elements of secondary structure as well as in whole protein. Most proteins are quite compact, and closely packed protein interior generally exclude water. The tertiary structure of a protein is determined by its primary structure. What is primary structure? It is the sequence of amino acids in the polypeptide chains. So first of all, the polypeptide chain is folded to form secondary structural elements. Alpha helix and beta sheet are the two principal secondary structural elements found in proteins. These secondary structural elements are associated in specific pattern with the help of different types of loops and turns to produce the three-dimensional structure of a protein. This three-dimensional structure is the tertiary structure. This figure shows the tertiary structure of a protein. As already mentioned, non-polar amino acids are generally found in the protein interior. Charged and polar amino acids are generally found at the protein surface. Polar amino acids are also present in the protein interior. In this figure, there are two alpha helices and this is the beta pleated sheet. So these secondary structural elements are connected with the help of loops and turns to produce the tertiary structure. The tertiary structure of a protein is stabilized by different types of interactions such as hydrophobic interaction, disulfide bonds, ionic bonds and hydrogen bonds. All these linkages impart stability to the tertiary structure. First one is hydrophobic interaction, which operate in the protein interior between non-polar R groups. Second type of linkage is disulfide bonds, which are formed between the R group of cysteine amino acids. Disulfide bond is a covalent bond. Third type of linkage is ionic bond, which is formed between oppositely charged R groups. And the last one is hydrogen bond. So with the help of all these types of linkages, different types of secondary structural elements in different proportions are organized together in a specific pattern to produce the tertiary structure of the protein. Each globular protein has a unique tertiary structure which contains a certain combinations of secondary structural elements. Proportions of alpha helices and beta pleated sheets and the order in which they are connected provide an informative way of analyzing protein structure. Supersecondary structures, also called motifs, are groupings of secondary structural elements. They are found in many unrelated globular proteins. Some of the commonly found supersecondary structures are beta-alpha beta motif, beta hairpin, alpha-alpha motif, and Greek key motif. The most common supersecondary structure is beta-alpha beta motif, in which an alpha helix connects two parallel strands in a beta sheet. Another common supersecondary structure is a beta hairpin, which consists of antiparallel beta strands connected by relatively tight ribosomes. The third one is an alpha-alpha motif, in which two successive antiparallel alpha helices are packed against each other with their axis inclined. And the last one is Greek key motif, in which a beta hairpin is folded over to form a four-stranded antiparallel beta pleated sheet. Major types of secondary structural elements such as alpha helices and beta pleated sheets occur in globular proteins in varying proportions and combinations. Based on this, proteins can be classified into three different groups. First one is alpha protein, which consists mainly of alpha helices. E. coli cytochrome B562 is an example of alpha protein. It consists mainly of alpha helices. Second one is beta proteins, which contains a large proportions of beta pleated sheet. Different types of immunoglobulins are examples of beta proteins. Immunoglobulins are devoid of alpha helices. However, most proteins including lactate dehydrogenase and carboxypeptidase A consist of a mixture of alpha helices and beta pleated sheet. Such proteins are called as alpha beta proteins. Alpha beta proteins. All these classes can be further subdivided into different groups based on their topology. For example, extended beta sheets often rolls up into beta barrels. There are three different types of 8-stranded beta barrels, two of which are all beta structures consisting of multiple beta hairpins. Whereas the third type can be considered as a set of overlapping beta-alpha beta motifs. So friends, this is all about the tertiary structure of proteins. For more updates, please subscribe my YouTube channel, Sun Biology Class.